You know, in the world of biology, we are living in an age of just unbelievable information. We have the blueprints for countless forms of life, but they're written in a code of billions and billions of letters. So the big question is, how do we even begin to read it? Well, that's what we're going to crack today.
See, it all starts right here. The cost of DNA sequencing has just absolutely plummeted. What used to take years and a fortune can now be done super quickly and cheaply. And the result? A data explosion. I mean, every single day, our digital library of life's code, DNA, RNA, protein sequences, it's growing at a mind-boggling rate.
And that leads to the central challenge, right? Just having all this data isn't enough. It's kind of like having a library with millions of books, but they're all in a language you don't understand. We need a tool, a method to search through all that noise and find the meaningful passages, the really crucial instructions hidden deep inside.
And that tool? It's called sequence alignment. This is the fundamental procedure that lets scientists compare these biological texts. It's how we find relationships, predict functions, and ultimately, it's how we begin to understand the story of life, written in its own language.
At its core, the idea is incredibly simple. Imagine you have two sentences. You'd line them up, right, to see which words are the same, which are different, maybe there's a typo, and where a word might be missing in one but present in the other. Well, biologists do the exact same thing, just with sequences made of genetic letters. So it all breaks down into three simple ideas. When things line up perfectly, that's a match. When they're different, it's a mismatch. And when something is missing from one sequence, we call that a gap. These three concepts, matches, mismatches, and gaps, are the absolute foundation of sequence alignment. Okay, so let's apply that same logic to a real biological example. Now, this might look a little complex, but I promise the rules are identical. We're aligning two protein sequences. The vertical lines, those show a perfect match. Where the letters don't line up, that's a mismatch, and you see those dashes? Those are gaps, and they probably represent an insertion or a deletion that happened sometime over evolutionary history.
So why do we even see these similarities in the first place? The answer is homology. It's a fancy word that just means when two sequences are very similar, we can infer they share a common ancestor. This can happen between different species, those are called orthologs, or it can happen within a single species, like when a gene gets duplicated, and those are called paralogs. It's all part of building this giant genetic family tree.
But it gets a little more nuanced, especially with proteins. Sometimes a single letter, an amino acid, can be swapped for another one that's chemically really similar. Think of it like swapping a screw for a nail that's the same size. The structure might hold up just fine. This is what we call similarity. It's not an exact match, but it's a change that doesn't break the machine.
Then, on the other hand, you have identity. This one's the most straightforward measurement. What percentage of the letters are an exact one-to-one match? So here's the key point. For proteins, similarity and identity are different things. But for DNA, which only has 4 letters, they're basically the same thing. An A is an A, and that's it.
Okay, so we know what we're looking for, but how we look for it really depends on the question we're asking. Scientists use two main strategies for this: global and local alignment. Global alignment is when you try to match two sequences from start to finish. Think of it like comparing two entire novels to see how similar they are overall. It's great for sequences you already know are pretty closely related. Local alignment, though, is more like a search for one specific identical paragraph that's hidden within two completely different books. It's perfect for finding small, really important shared regions in sequences that might be very different otherwise. All right, so now that we've got our strategies, what are the actual tools we use to perform these alignments? We're going to look at two major methods, one that's very visual and one that's just pure computational power.
First up is the dot matrix, or a dot plot. This is a wonderfully simple and visual way to compare two sequences. It's really just a graphical eyeball test for finding regions of similarity. And here's how it works. You put one sequence on the x-axis and the other on the y-axis of a grid. Then you go through and just put a dot in any cell where the letter from the row and the column are the same. When you're done, you just look for patterns. A long, continuous diagonal line of dots, that means you found a strong region of alignment. A break in that line? Well, that signals a potential insertion or deletion. Simple, right?
But what if we need to find the single best possible alignment, mathematically speaking? For that, we turn to a much more powerful engine, dynamic programming.
This is the powerhouse behind most modern sequence alignment. So instead of just placing dots for a match, dynamic programming builds a scoring grid. Imagine it gives you points for every match, but it takes points away for every mismatch or gap. The computer then calculates every single possible path through that grid and finds the one with the highest total score. This guarantees the best possible alignment. This is the powerful method behind famous algorithms like Needleman-Wunsch for global alignment and Smith-Waterman for local alignment.
So, we have these incredibly powerful tools to compare sequences. But let's circle back to the big question. Why does any of this matter? What is the real-world payoff of being able to read this code?
And this is the payoff. By aligning sequences, we can pinpoint the exact mutations that cause diseases. We can build evolutionary trees that show how all life is related. We can find these conserved patterns that tell us a new, uncharacterized protein belongs to a certain family, which then lets us predict its structure and function. This knowledge is absolutely essential for designing new drugs and better medical diagnostics. It is truly a cornerstone of modern biology.
So we have this ever-expanding library of life and the tools to read it. Sequence alignment is our Rosetta Stone, allowing us to translate all this genetic code into real biological meaning. The only question left is, with so much still to discover, what vital secrets will we choose to read next?